Thanks a lot. Is this microphone going to work okay? Great. So I think we have till 8 o'clock with time for questions built in. Is that right? And with that in mind, I wanted to start not with my typical slides, but with a preface, which provides some context for why I've become very interested in engineering biology. And without any apologies, I'm just going to jump into this. This is meant to be a little appetizer, if you will. And if it's not technically accessible, hang tight, and you'll get back to the normal introduction and see what's going on. So when I was a student back in the mid-90s, I started as an engineer coming into biology working on viruses, not because I was particularly interested in viruses, but because of all the natural biological systems in the world, they're relatively modest in terms of the number of components and what have you. And so here are some different diagrams of viruses, from micrographs to sketches. And the particular virus I was working on is called T7. And this is a virus that infects bacteria, E. coli, that live in your gut. So what you're looking at here is a movie of E. coli growing, and they've all been infected with a virus. And so you see they just all popped. You can't see the virus because the virus is really tiny. The E. coli are about 1 micron, so much smaller than a millimeter. And what's interesting about these particular systems, not if you're a scientist, but if you're a technologist, is that they build themselves. These particles make themselves. They can copy themselves. They're tiny, so they're nanoscale. And all that means is if you think about computers being really tiny, you can do lots of important things on a small amount of space with a tiny object. And you can program these objects by manipulating DNA. So those are just some interesting features that make these viruses of interest. If you go onto the internet, you can find databases which will give you the DNA programs for viruses. And we can come back and talk about that later because that might sound scary, and in fact, I think it is sometimes. And in this case, I'm looking for bacterial virus, and I look up a particular virus, this virus T7. You can see it says that right here. And the genome of T7, the DNA is the genetic information that encodes all the material that programs this virus, is 39,937 base pairs long. A base pair is a letter of DNA. So if you download the sequence, there it is. Right? And you can just make out the A's, T's, C's, and G's. Right? Now, a different level of representation or depiction is shown here, where these little cartoon blocks represent 10 or 100 or 1,000 of those letters, and they're organized left to right. And this is the way a geneticist would describe the architecture of this virus's DNA, where you have different proteins encoded 1, 4, 5, 6, and so on, and then all these regulatory elements. And so this is what I encountered as a young engineer coming into biology and trying to learn from the genetics community, hey, what do we know about these viruses? I should say, I'm probably going to talk at this pace, but if you want to interrupt me, throw something from outside and get me to ask or answer a question, please interrupt, okay? All right, believe me, please do that. That'll be good. So this is one ordering of these natural elements. Here's another ordering of the elements. And you can imagine having a deck of cards and shuffling a deck of cards, how many different ways there are to order the cards. So it is with these components on the virus's genome. You could have this ordering or da-da-da-da-da, many, many different orderings. More orderings than there are the number of atoms in the universe. And one of the things that was interesting to me is why does nature, when we isolate this virus from nature, why does the virus have this particular ordering, the one we find? And a nice way to answer that is to try out other orderings and see what happens. Now the problem is because there's so many orderings, there's actually no way to try them all out physically. Does that make sense? There's just too many. So the approach was, if I don't know what this program's going to do and I can't build them all, maybe I can build a computer model of how this genetic program's going to operate. And then I'll run the simulation of this particular genetic program on the computer and see what the computer says it will do. Sort of like with varying degrees of success, we have computers that will help us simulate what the weather might be doing. So this is I won't go through the details of this, but this is the world's best simulator we've ever had for simulating the infection of a cell by a virus, where we're tracking the individual molecules running along the DNA, reading out the DNA, making proteins, doing all the things that genetic programs do. And it turns out that those programs aren't very good. that when we make predictions on the basis of these models and simulations, say, I think I could redesign this genetic program to make a virus that grows faster than the natural isolate, every time I've actually gone into the lab and tried to construct that, I would make something that actually grows slower. So my predictions, based on a very simple natural biological system, which has been studied for now 60 years, my predictions don't work out so well. And as an engineer, when the bridge falls down or the plane crashes, you're not supposed to sweep that experience under the rug. You're supposed to collect it as a failure and then do what's called a failure analysis. And so when my computer models didn't let me make accurate predictions of the behavior of natural biological systems, I did what I was taught, which was a failure analysis. And so here's one of the things I noted. This is the table of all the different genes on this virus. A gene is a region of DNA that makes a protein that does something. So you could have a protein that encodes the shell of the virus, where the DNA is packaged. Or you could have another protein, which is the tail fiber, where the virus lands onto the cell. These genes that are highlighted in this reddish magenta color, those are the genes for which we have no idea what they do. It's like lifting up the hood of your car and looking at something under the hood and go, what the heck does that do? And you don't know. But in the case of a car, you can go talk to your mechanic, right? And if your mechanic doesn't know, you get a new mechanic, or you go talk to the people who designed the car. And somebody should know. In this case, because it's a natural biological system, when we lift up the hood and we see that 40% of the components, the ones that are highlighted here, we don't know what they do. There's nobody to go talk to. So as an engineer trying to model this thing and predict what happens when I change it, this is a nightmare, because you don't know what 40% of the components do. That surprised me. Here's the next thing that surprised me. When I went to go look at the DNA sequence itself, not these abstract components, but the actual thing that runs inside the cell, here's an example of what you find. A, C, G, C, A, A, G, G. So this one gene, which is given the number 2.8, comes in from the left, and it's coded by this DNA, and then it stops at this red TAA. The next gene, gene 3, starts over here at this green ATG and goes off to the right. So you can immediately see that these two genes share some real estate. You know, it's like they're neighbors, and they each think they own a piece of property on the DNA. I have no idea what this means. I don't know if this is a form of data compression, that the virus compacts its DNA by figuring out how to overlap the coding sequences for these two genes, and so it's a little bit more efficient because it has to fit into a tight particle. Or I don't know if this is some form of regulation that controls the reading out of the gene. Nobody's really looked at this before. This turns out to be problematic because, again, if I don't know what this architecture does, I can't model it, I can't simulate it, I can't figure out how to predict what will happen if I change it. Around this time, I became aware of other people who were studying evolved systems. So this example DNA program I'm giving to you comes from nature, and so we believe it to have been evolved. And so when we look at the sequence, if it doesn't make sense, well, maybe we don't understand enough, we can't make sense of it, or maybe it wasn't evolved to be sensible, if that makes sense. So for comparison, here's another type of evolved system. This is a diagram for an electrical circuit. Now, this electrical circuit was not designed by a human designer. It was designed by a computer program that can simulate the evolution of electrical circuits. So that sounds a little complicated, but that's how it is. And so this electrical circuit was evolved to do something. Can anybody guess what this circuit does? It's supposed to take the square root of the input voltage to the circuit. Now, I can't explain to you how it works. I can't explain to you why it works, and I can't explain to you why the simulated evolutionary process produced this particular design. But it was a very exciting question to me answering these sorts of things when I first saw this, because I figured if I could understand how evolved electrical circuits work, then maybe that approach would help me understand how evolved DNA circuits work. So I went to talk to Jerry Sussman, who's one of the best electrical engineering professors at MIT. And I was really, really excited. I brought the schematic and I said, Jerry, how does this circuit work? And he has very good eyesight. So the first thing he said was, your printout isn't very good. And then the next thing he said, pretty quickly, was, where did you get this from? Who designed this? And I said, oh, this guy, Koza out in California, he evolved this circuit. He evolved it. Thought about it for 5 or 10 seconds more. He says, I refuse to consider your questions. I said, what's going on? He says, you're asking me to waste my time. This thing was not optimized to be easy to understand. It wasn't optimized to be easy to explain to you. Really intelligent design would have documentation. So taking these ideas back to biology, given this sort of circuitry, the question that came up was maybe we should do an experiment. Maybe we should consider designing and building a new genome. Whereas if the natural one has many things we don't understand, perhaps we could throw them out and just see what we could learn about how the natural programs work by changing them. So for example, we could take the natural piece of DNA on the top and find the things that we think are beautiful about it, like the coding regions for these different genes, and things we don't understand, like the overlapping of the genes, and just decompress it. Give each piece of DNA its own function, and then make some changes so that each piece of DNA only does one thing, right? You don't want a system where, say you're driving your car and you adjust the volume of the radio and the steering wheel starts to move. Right? We'd like to separate the functionality. And so we tried to do that, and we designed a new genome that doesn't change any of the proteins. It just changes the DNA architecture. We were very worried that when we did all this work, we would make something that doesn't grow at all, that we couldn't even study. And so we did that. We made about 600 changes to the genome. And the big result for us, without getting too much into the details, was the resulting engineered genome was viable, meaning it could infect cells and grow and divide. And that was a big result for us because it said we could make 600 simultaneous changes to the DNA of a natural organism and still get something that was viable. Now, if you understand what's being shown here, these are basically Petri dishes. These are 10-centimeter dishes, and they're bacteria growing on it. And each little circle is what's called a plaque. So a virus starts out in the center of those, and it starts growing and dividing, eating away at the bacteria. And over time, it diffuses out and eats away more and more bacteria. And the bacteria, as they go away, there's a black background behind the plate, and so you see this black circle. If that doesn't make sense, you can think about being up on a satellite, looking at a forest, and a lumberjack is parachuted into the forest and starts chopping down trees. And every time a tree gets chopped down, 100 new lumberjacks come out and chop down more trees and eventually get a clearing in the forest. That's what you're looking at, except the lumberjacks are little virus particles and the bacteria are the trees. So on the left, you see what the virus does from nature. It makes these big clearings in the forest of bacteria. This is our engineered virus. And the clearings are much smaller. So that shows that it doesn't grow very well. Again, the big result was we could make 600 changes to a natural piece of DNA and produce something that wasn't dead. That was a big deal. And we wrote it up, and we were pretty happy to have a paper. So that gets me to my bigger topic, which is the idea of if we wanted to have a biology that we could understand, that we could predict what happens when we change things, and those ideas themselves might be controversial, wanting to realize that future, maybe we should build it, instead of trying to reverse engineer and refine and perturb natural biological systems. So with that preface, I'll now move into the primary part of my talk. So it turns out that part of this work involved making a comic book. And the comic book was trying to explain to folks why you might want to set out, or rather how you would go about writing DNA programs, engineering DNA. And so in this case, there's a teacher and a student, and the student's excited to see that there's this stuff called life out there, and it's all running on DNA programs to a first approximation. So if you could engineer DNA programs, maybe you could make something new. So we're going to try how it might work. How do these things work, these biological things? Well, it turns out they have programs made of DNA inside them. And so if you could go in and look at the DNA program, this is what you might see. And of course, you'll note that nobody really understands, as I just noted, how these natural DNA programs work. We don't know what all the components are, for example. Nevertheless, this brave young student decides that I'm going to go try and learn how to do this. What might he do? Bacteria balloons. I'm going to program these little bacteria to make balloons. This teacher is a little bit skeptical. I guess nobody's done that before. But all I really need to do, the boy says, is get them to form a film and then start producing hydrogen gas. And so how would I do that? Now here's where the story gets a little bit fictional. First, you assemble the DNA parts that encode your program. You get the parts from a catalog. One gasomatic module, one balloonogenesis module. Cool, here they are right in the catalog. Then I'll load the DNA, shown not to scale here. I'll load the DNA into our bacteria. And what does the bacteria think about this? The bacteria has some opinions. The program inserted into the cell starts to run, much like loading a new computer program onto your computer. The bacteria is doing something, looks pretty good. It keeps going. That doesn't look so good. There's a little bit of a problem, and it turns out that the student forgot to tell the program to stop, which is a good thing to learn in a laboratory setting. Maybe you want to learn a little bit more before you try this again, and so on. Well, it turns out that a couple years ago, we started making a catalog of standard biological parts. It's called a Registry of Standard Biological Parts. We have no idea how to do this, but we figured the only way we'd learn was to try it out. And today, there are over 2,000 pieces of DNA, standard DNA parts called biobrick parts, that are freely available to students. What sort of parts are there? Well, here were some of the students who were making parts last year. And in particular, these were some students from Melbourne, Australia. And these students from Melbourne, Australia followed a technical standard invented by Tom Knight at MIT, and they made biobrick part I750016. Now that number shouldn't mean anything. It's just an index number. But this is a gas vesicle polycistronic gene. That's a complicated way of saying this is a protein balloon. Somehow, a couple years after the comic book got written, some people on the other side of the world made a biobrick part that makes a protein balloon. Huh. Moreover, the scientist upon whose work they had made their protein balloon, they were actually at the University of Massachusetts, right next door. So I was really embarrassed that I didn't know anything about this biology, and it had been done in my neighborhood, and that these folks on the other side of the world had made a standard biological part that we could readily use. And so if you go into the database online, there's the DNA sequence encoding a protein balloon. Now this protein balloon isn't 1 meter across. It's 60 billionths of a meter across. So it just fits inside a cell. And so what that means practically is if you take this DNA and you put it inside a cell, and if you change the number of protein balloons that get assembled, if you have a lot of the balloons, the cell will float in a liquid. And if you don't have a lot of balloons, if you have none, the cell will tend to settle towards the bottom of liquid. So this isn't rising or sinking in the air. It's rising or sinking in a liquid, which is interesting if you're doing, say, a fermentation process or making beer, and you'd like your yeast or what have you to end up at a particular place. Huh. What other sorts of things can you find in this collection? Here's BBA J45200. Again, this number doesn't mean much. This turns out to be a banana odor generator. What would you use that for? Here's the DNA. You put this DNA into a cell, and the cell makes a chemical, isoamyl acetate, that smells like bananas. It turns out this piece of DNA had been made by a bunch of first and second year undergraduates at MIT. And they hadn't yet taken a laboratory course, and so when they came into the lab that summer, one of the first things they noticed was E. coli doesn't smell so good. And so they decided they wanted to reprogram the bouquet of E. coli. They called their project Eau de coli. And their project was somewhat sophisticated. They didn't just want the E. coli to smell like one thing or something else. If the cells were growing, this was the genetic program they wanted to write. And you'll notice there's no DNA sequence on here. It's a very high-level language. If the cells are growing, call wintergreen, which is methyl salicylate. Otherwise, if the cells are resting, call bananas, which is isoamyl acetate. They took this program and they compiled it down to an intermediate level of technical description. So you can go from some chemicals inside the cell, charismate and leucine, through some conversion steps to get to salicylic acid and eventually methyl salicylate and isoamyl acetate. So they were able to sketch this out pretty quickly just by looking in the various databases and tables of biochemistry. And then they went to start to assemble the DNA program. And they did not get this entire DNA program working in one summer. What they were able to get working was they were able to get this section here and this section here, where they could get banana smell produced during resting stage and wintergreen smell during growing stage. And to get that going, that means they have to provide this intermediate chemical. That was so cool that they showed up at an event with live cultures and asked people to smell to see if it worked. And it turns out that most people, almost 90% in their test, could agree that E. coli are stinky, right? The mint did not work so well. Only about 2/3 of the folks could identify the producer of the methyl salicylate chemical. And most people could figure out that banana smelled like banana. Not bad for 18 and 19-year-olds who are doing a genetic engineering project for the first time. So what we talked about so far are things like this. Actuators, output devices. There are obviously other things you'd like to be able to engineer with DNA, right? And if you look at this robot as an example, you would quickly notice it has eyes, right? Some sensors or input devices. And so another example you might want to look for in the registry is of input devices. So here's a part, M30109, which is a light responsive system. This is a primitive eye. Again, this is going to work inside bacteria. The E. coli that live in your gut are typically in the dark, so they're not naturally directly responsive to light. And so by adding this part to E. coli, you can have them have a programmable light sensor, if that makes sense. So this is work that was done by Chris Voigt in San Francisco. And practically the way this works is you spread the bacteria with this light sensor on a plate, maybe 10 centimeters across, and then the bacteria will produce an enzyme that causes the plate to change color. So it's a primitive photography system. The exposure time is about 4 hours. The bacteria, though, are a micron. So you have almost a billion of them per square inch. So it's like gigapixel per square inch resolution. Not only is it dense resolution, but these pixels are living things that can copy themselves and make whatever you can program them to make, like balloons or who knows what. So I don't know if you can make this out. This is one of the first programs they made. It's just a film. The bacteria aren't self-differentiating into this pattern, but they're just capturing an image projected on it. And it says, "Hello, world," a very classic first computer program. Here's a bacterial photograph of Einstein on the right. Here are photographs of Darwin in celebration of the series here, perhaps. Andy Ellington, a biochemistry professor. You could take photographs of your teacher on these primitive Polaroids, they're called. Here's the Hong Kong flag, Mother Teresa, the flying spaghetti monster, and so on. Okay, so what the heck is going on? What is up with synthetic biology, as it were? When I moved to MIT back in 2002, there was not a department of biological engineering. And so one of the items in my job description, as you will, was to work with more senior colleagues there, Doug Laufenberger and Linda Griffith, to help start a new department of biological engineering. And as I started working on this part of my job, I got confused. And I got confused because when I went to the library, I found this paper that had been written in 1939 by the president of MIT. And the paper says, "The Genesis of a Curriculum in Biological Engineering." So how is it that in 2002 I could be hired at MIT to help start a new program in biological engineering? That means that whatever happened almost 70 years ago crashed and burned, right? Or didn't work. And remember what I was saying before about when the bridge falls down, you want to pay attention to that. So this was really troubling because not a lot of people were talking about this. And if you get the paper, hopefully I can bring up some details from it. Here's a table from that paper. It begins to show the curriculum. And I'm only showing the first 4 years. Normally you think about going to college and it's 4 years. This program was actually 5 years long. And maybe that's why it didn't work. But you got a degree in bioengineering and in biophysics. And that's impressive because at the time, 1937, 1939, that's when the Rockefeller Foundation was beginning to make investments in the physics community, helping physicists to come into biology. And that's in part what gave birth to what we know today as molecular biology. Folks trying to understand life at the level of molecules and atoms. This is before the structure of DNA was worked out in the 1950s. And so a very important time in the history of biology, right when this work was taking place. Now, I don't know why this project didn't work almost 70 years ago. And it's something I'm still trying to figure out. Because I don't know the answer to why that project didn't work, I have to replace the process with a surrogate, if you will. And the surrogate is the following question. And this will be the second part of my talk. Here's a question for your consideration. If you guys are from Notre Dame, so if one of you were to go to MIT, or all of you were to go to MIT next year, right, and decide what you're going to major in, if you were to choose to major in biological engineering, right, this is a question for everybody, but you have to actually answer it, right? If you were to choose to major in biological engineering today, what would you expect to be able to do after 4 years? Right? So because I can't tell you why the program from almost 70 years ago failed, this has been my replacement question. To help you think about that, the next part of my talk is a bunch of postcards. These are postcards from around the world showing what the engineering and scientific research communities have been able to do with biology. And many of our students coming to MIT have some awareness, not of all these postcards, but of some of these postcards. And so again, this is going to come pretty fast, and it's just meant to give you a sense of what is technically possible today. This is a postcard from Japan, from Mitsubishi. And I didn't know Mitsubishi was working on this, but what they did was they took the genome of one organism, bacterium that lives in the ocean, and they moved it into a second bacterium, making a new bacterium that has a composite genome, all the DNA material for almost the entirety of 2 bacteria. What's notable about this project is just the scale of manipulation of DNA. They were able to make a composite genome that's 8 million base pairs or letters long. The first project I was telling you about, the virus, that's only 40,000 letters long. You can make a piece of DNA that's 8 million base pairs long. That's almost as big as baker's yeast, which we depend on for bread and beer and what have you, right? That baker's yeast is about 12 million base pairs long. Here's a project that got a lot more attention recently. Folks built a piece of DNA that's 600,000 letters long, but they made it from scratch. They made it from sugars. What if you could build whatever genome you wanted? Here's a postcard from France. And again, I'll try and explain these. This project freaked me out when I saw it. Apparently, our ancestors, about 5 million years ago, were infected by a virus. Now, I don't know what was happening 5 million years ago, but this virus that infected our ancestors inserted its DNA into our genome, into our genetic material, and then went dormant. And the DNA of the virus got copied about 24 times. And then over a long period of time, the dormant virus genomes hidden out inside ours fell apart. They sort of rusted. They broke down. But they all broke down differently. And so if you look at the sequence of the human genome, you'll find a couple dozen copies of this ancient virus. And not one of them works. But because they all broke in a different way, you can infer what the ancestor looks like. It's like figuring out how a car works by taking 100 different broken cars that have each broken down in a different way, and then figuring out what the union of the working parts of the car are. It sounds complicated. They used some computers to help. And then they built the inferred sequence, the putative, the expected ancestral virus. And when they did that and put that back into cells, they found that, in fact, they were able to make a fully infectious virus. So this is like the movie Jurassic Park, except you don't need the physical sample of amber. You know, what is there? There's a mosquito inside the amber that's been a dinosaur, so it's got dinosaur blood. So you get the mosquito out of the amber, you extract the dinosaur DNA. You don't need any of that. You just take the sequence information that's already inside all of us, you do some computational analysis, and you infer a virus from 5 million years ago, and you build that. That's crazy. Well, the first time I heard that. Here's a postcard from France. Normally you think of France, you think about baguettes. Another movie reference. There's a movie called Fantastic Voyage. Somebody's fallen ill. They need to be fixed. The idea is to take a submarine with a bunch of doctors inside it, shrink it down, and then the submarine with the doctors inside it will go fix the patient. That is a really, really good idea, except that the physics community has not provided shrinking rays. And so if you wanted to provide a practical surrogate for a Fantastic Voyage, what you might choose to do is take something that's already really tiny, say a bacteria. Normally you think of a bacteria as being problematic, but what if you could reprogram a bacteria to be a little programmable submarine that goes inside and finds a problem and tries to fix it inside you? And so here a lab from Berkeley and San Francisco collaborate together to implement some genetically encoded sensors that can sense the environment around the cell. They have some logic, and they have some actuators, and they're trying to build bacteria that will identify and attack tumor cells. This is a paper that just came out last week, a postcard from Los Angeles. And basically what they've done, Christina Smolke's lab, she's figured out how to implement logic, like and, or, and so on. Computational logic out of not DNA, but RNA. So it goes DNA, RNA, proteins. And so these little RNA computers aren't ever going to replace silicon, in my opinion, but what they're likely to do is get us access to computation inside living cells where we can't put silicon. And so that's a new area for computing to take place. Here's a project from San Francisco where folks are trying to make a drug for treating malaria. Malaria isn't a problem in the US so much, but in other parts of the world it's a problem. And the cost of the drug called artemisinin is too much, too expensive for the folks who have malaria. The sourcing of artemisinin, as I understand it, is from the bark of a tree called the wormwood tree. What if instead of growing the trees, harvesting the bark, and extracting the chemical, you could reprogram yeast to make the drug for treating malaria? So Jay Kiesling's group has collaborated with a bunch of folks to get this to work. Here's a project that turned into a company coming out of MIT. A lot of times you'll think of a power plant burning coal or oil, and it will produce a lot of carbon dioxide and other compounds, nitrogen compounds in the smokestack. What if you could put algae, which like carbon dioxide in the presence of light, they fix the carbon dioxide and make a fuel, an oil, basically. What if you could recapture the carbon coming out of the smokestack of a power plant and clean up the off gases while getting some more fuels? So that's the idea here. The biology on this works pretty well. The engineering problems have to do with scale up and getting access to the water and the land. So a very interesting idea that's a little tricky, but yet another postcard. Here's a paper. I think this was it's from California and Maryland. Mice apparently see in 2 colors. These researchers added a 3rd photoreceptor to mice so that the mice can apparently see in 3, whatever that would mean to a mouse. Here's the paper we talked about already. You add a single photoreceptor to bacteria. The bacteria can take primitive photographs. Here is a paper from Princeton where folks are programming bacteria to differentiate into patterns in space. So if you eventually wanted to be able to program the formation of a tissue, you would need to figure out how to write a program that differentiates in space. And this bullseye pattern is a first example of genetic programming that lets you make patterns in space. Here's a project from Los Angeles, from Caltech, where folks have invented a language for programming DNA, which normally you think of as an information carrier, but here they're actually using it as a physical material. They figured out how to program DNA to fold up into any 2-dimensional pattern you want within the bending limits of DNA. So you could get a star, a happy face, the world map, and so on. What's cool about these patterns is they assemble themselves. You don't have to put everything exactly like a jigsaw puzzle. This thing goes, right? Here's some things that we can't do. So these are the last 2 postcards. This is a sponge that lives in the ocean. And it has a skeleton made of inorganic materials that have interesting optical properties. So Joanna Eisenberg, she was working at Bell Labs. She's now at Harvard. And they were interested in understanding how the different components of the skeleton behave in response to the mechanical properties and the optical properties. This thing on the left is the skeleton of the sponge. It's about 10 centimeters tall. This thing makes itself. How would you do that? Somehow biology can assemble things that look like this. Last postcard. This is something we can't do as well. This is from the architecture department at MIT. I was walking by it one day, and I noticed this thing down in the bottom left, which the students had called Fab Tree Hab. And apparently what they'd like is they'd like to be able to have gigantic programmable gourds that differentiate into 4-bedroom, 2-bath houses. You think about the gigantic pumpkins that come up this time of year, this would be bigger. Now what's interesting is over in France again, some folks had figured out how to shape vines so that over time you could get these primitive 2-story huts. And so that's not quite what is shown on the left. And obviously we'd have to figure out how to cross redwood trees with bamboo and giant gourds, to get this to go. But maybe it's plausible. So question. If you were an 18-year-old first-year undergrad at MIT, what would you expect to learn after 4 years of biological engineering? That's the question that's pressing for me, because if we can't figure out how to answer this, then 70 years from now, somebody else is going to show up and have to help start a new department of bioengineering. Well, here's what I've heard from the students. I'd like to learn how to design and build living organisms that behave as expected. I'd like to learn how to write DNA programs that do my bidding. We don't know how to teach them how to do this. But it's this set of expectations which, for me, has driven a lot of my own research interest. And that's, for example, what led us to begin to make something like a registry of standard biological parts. Now, the reasons they want to do this are old. Not old in a geological sense, but old over the last 40 years. So when biotechnology was first developed, people immediately wanted to use it to make drugs, insulin for treating diabetes by moving the gene for insulin production into bacteria. That's worked out pretty well. Gene therapy, that's not worked out so well. Nitrogen fixing crops, corn that doesn't need synthetic nitrogen fertilizer, that's not worked out so well. If you look at the 3 big categories of promises, 3 of them at least, from the first generation of biotech over the last 35 years, we've really only delivered on 1 of the 3. And of course, you could imagine many other applications of biotechnology, right? So you could have global energy cycles, carbon fixation. You could be worried about where phosphorus is going to come from. That Eau de Coli project sounds like a stunt, but all of a sudden you could use odorants as reporters of cellular state when you can smell things but you can't sample them. Or if you wanted to be a science fiction author, but not too much, you could imagine engineering the ecosystems on your skin to get distributed manufacturing of scents and fragrances by loading up new DNA programs instead of having a centralized facility and sending a lot of money over to Chanel. So what if we wanted to make all of biotechnology come true? The constructive aspects of it, at least, and not at some notional point in the future, but say, I don't know, 30 years from now or less? What would we do? How would we set out to take this natural substrate and make it more engineerable so that if this is actually possible ever, it happens? Well, to go into the last part of my talk, I want to explain where some of the basic engineering ideas are coming from and sort of what the state of the art is, and end with some questions. So where these ideas we're trying out right now, where they're coming from, is from history. And basically what we're doing is we're being amateur historians or thieves of other technology transitions in human experience. So I'll give you 2 examples. This one is from what I'm familiar with, civil engineering. A long time ago, at the beginning of the Stone Age, we humans decided that rocks are useful. You can build a wall or a dam or something like that. But the rocks we find in nature are not easy to use. They're all different. And the scientific community can go study the rockiome, and that's great. Characterize how each rock is different, but there's a complementary activity which is making rocks easier to build with. And so many folks will be familiar with these things, quarries. This is a radical invention of technology in the Stone Age. You're now making rocks that have regular interfaces. so they become easier to build with. Things like flying buttresses and cathedrals become possible, but some of the first cathedrals take decades, if not centuries, to make. Later on in the Stone Age, more akin to what we're familiar with today, folks become dissatisfied with rock, so they grind it up and they make new synthetic rock, which is called concrete. And once we have concrete and many other tools like computer-aided design, you see astonishing artifacts like this. This is a bridge in France that only took a couple years to make. And if you can make things like this in a couple years, you can make skylines like Chicago or here, Hong Kong, in a very small amount of time. So that's an example of a technology transition, one that took thousands of years. The second example will be one that only took 60 years. So this is a schematic, a photo rather, of an electronic computer just after World War II. Around this time, people like John von Neumann are building computers in the basement of the Institute for Advanced Study at Princeton, and the contract paying for this is to help with the design of hydrogen bombs, right? Not the most constructive thing. Somehow, within only 25 years, people are making computers that look like this. And I think it's because people thought that computers were cool, and they were fed up with limited access to computing. That only is 25 years. That's not thousands of years. And then within another 30 years, we have this world, web-based computing, the iPhone, and other curious artifacts that are somehow appearing. So how come this is true? Well, one thing that drives technology development is the applications of the technology. But that doesn't really give you these big revolutions. I would argue that the big revolutions come from the tools that help with the process of building. And that, to me, is what synthetic biology is really about as an engineer. How do we develop the tools for biotechnology? Here are the tools that were used around the computing and information revolution of the last 60 years. The theory of information, device design, computer languages, standardization, abstraction, fabrication of silicon wafers that the computers get built from, control and I'll show you an example. This is an example of a system that's growing itself. And this sounds like a biological example. This is actually an example from computer science. Computer scientists, oftentimes, the great computer science researchers will invent new programming languages. And Java, C++, these are all programming languages. These programming languages control the operation of machines in time. Some of the most daring computer scientists today are writing new types of programming languages that don't control machines in time, they control machines in space. What if you could write down a program that operates inside a machine, and as the machine copies itself, the program differentiates and assembles into the 2- or 3-dimensional structure that you want? Kind of like that gigantic programmable gourd I want. I need somebody making languages for programming space. So this is work done by Radhika Nagpal. She's at Harvard. And she's figured out that you can basically program these reproducing machines to make circles and then stop, and then at a particular point on the perimeter, grow another circle and repeat. This is one of the first languages I've ever seen for programming space. You could probably come up with a better language. And there's a lot to learn from developmental biology and embryology, right? Absolutely. So if I go back to biotechnology and look at the last 35 years, these are some of the underlying tools. Recombinant DNA. You take these little molecular scissors and you can cut and paste pre-existing fragments of DNA. It's great, it's unbelievably tedious. Polymerase chain reaction lets you take one molecule of DNA and amplify it up so you have more to work with. Sequencing of DNA lets you read it out. These aren't the only tools we could make. We could have tools like automatic DNA construction. Push the button, get the DNA. We could simplify genetic engineering via abstraction and standardization. So I'll show you what this looks like today real quick. It's very naive, it's work in progress. It's competing with nothing to a first approximation, and so any success is having a big impact. Here's automatic construction of DNA. These 4 bottles on the left are chemicals derived from sugar, produced from sugar cane, A, T, C, G. Each bottle costs $250. These bottles are in the form that plugs into a machine called a DNA synthesizer, which when programmed by a computer will stitch together a piece of DNA from scratch. So you go from sugar to a piece of DNA from scratch. There's an example of the sugar and the machine, and you stitch together a piece of DNA like that. All right. So this is one of the coolest technologies I've ever seen. It's sort of like building silicon wafers for computers, except it's more important. because you're building, it's not that silicon is unimportant, it's just this is DNA, which programs the living world, so it's got to be pretty important. Here's something for everybody's consideration. So this is on the x-axis time going back to 1970, and on the y-axis to the power of 10, a logarithmic scale describing how powerful technology is. So this is my most technical plot. So bear with me. This first curve right here, this describes the number of features or transistors, if you want to think about it that way, on a computer chip. This is called Moore's Law, after Gordon Moore, one of the founders of Intel. Moore's Law for computing. Computers get better by a factor of 2 every 18 months or so. That's been true since 1970 and explains why we have laptop computers today. This curve in the middle, this is how many base pairs of DNA you can read out. So if I gave you a physical piece of DNA and you wanted to read the ATCG, this is how many letters you can read out in a day. And you can see it started out kind of low, and then it got better really fast. So just to put this in context, in 1995, that's when the first bacterial genome was read out, ever. In 2001, only 6 years later, that's when the first human genome was read out, ever. Now, it's not that geneticists got 10 billion times smarter during the Clinton administration. It's because the tools for reading out DNA got hugely improved over that period of time. We live in a world today, this plot doesn't go out that far, where you can actually buy your own genome sequence for about $300,000, which is a lot of money, but that price was just announced in the New York Times to be dropping to $5,000, right? And there's people working to make the human genome sequence $1,000. Again, this is because the tools for reading out DNA are getting better, and they're getting better in a way that would be described as exponentially, right? This curve here is the last curve here. This is how many base pairs of DNA you can make from scratch in short little sequences about 200 letters long. So it's DNA synthesis. And you can see we're not very good, but we're getting better. And 2008, it's kind of like 1995 was for sequencing. 2008, this year, this is the first year that somebody built a bacterial genome from scratch. What will the world look like in 6 years? I don't know. My guess is it's going to be a very different world than what we have today, that people will be able to build 10 million base pair fragments, if not much larger fragments of DNA from scratch. That's like building yeast, the DNA of yeast from scratch, from information and sugar. What would you do with that technology? Okay, well, one of the things you realize pretty quickly is it's hard to program cells at this level, the DNA level, because you have to learn all this sorts of information. TAA, TA, CGA, CTC, ACT, ATA, GGA, GA. Obviously, that's a consensus promoter for the T7 RNA polymerase, a little element of DNA that drives transcription, and you can memorize these sequences by making them the computer passwords you use to log in. But that is a very tedious thing to do. And it would be akin to programming a computer at this level, the machine language of the computer or the machine language of the cell. It's not that this is a bad thing to do, but very few people today program computers in zeros and ones. So what we'd like to do, and we don't really know how to do this, but we're trying, is invent a programming language, higher level languages for writing DNA programs. And so we're going to make this up, and I'll do this quickly. We're going to call these things parts, and we hope to standardize them so they're reliable when you reuse them. And then we'll put parts together to make higher level objects called devices, which might implement Boolean logic or be protein balloons or what have you. And then we'll put devices together to make systems and da-da-da-da-da. So looked at a different way, we'll have what a computer scientist would call an abstraction hierarchy, where somebody at the top might be an expert systems designer. And they might think about how to reprogram the bouquet of E. coli, but they wouldn't have to know that DNA is made up of 4 bases, let alone anything about phosphoramidite chemistry or how to build DNA. That would be a radically different future than the one world we have right now, where everybody who's doing genetic engineering work really needs to be expert across all these levels. How can we do this? Well, we can take the natural component tree. For example, a bacteria that lives in the squid has a cell-cell communication system, and this helps the squid go hunting at night because it coordinates the production of light. Maybe we could take that biology and just extract the cell-cell communication module, do a bunch of engineering work, and wrap it up as a black box or a device that you can get off the shelf. So here, for example, would be a data sheet. We published this just this summer. And it's our first formal data sheet describing F2620, which is a genetically encoded receiver. It's a piece of DNA you can put into a cell, and the cell will boot up a cell-cell communication receiver device. So you could send a little sugar signal from outside the cell and have that program protein balloons or banana production or whatever you like. Again, we don't really know how to do this very well yet. This is a research paper from this year. Nevertheless, as we've started doing this work, as we've started making this pilot project, this registry, it comes back to the expectations of our students. And we're pretty honest with them, meaning we're no good to first approximation, and we'd like them to work with us. And so they've started helping. So here, for example, are 16 students at MIT back in 2003. And they were trying to make little primitive genetic clocks. And not one of their clocks worked. But when we tried to go get their DNA built, we ran out of money. And so we had to figure out how to get them to reuse their components. And that was what gave birth to the Registry of Standard Biological Parts. It was a way for them to share their parts. And that saved us $40,000 on DNA synthesis. That was so much fun that we taught the course again in 2004. And these students were inspired by that bullseye pattern that had been programmed into the bacteria. And so they were trying to program polka dots into lawns of bacteria. And not one of their projects worked. But that was so much fun that we didn't even wait a whole year. We ran the course again that summer, and we expanded to 5 schools. And you see the students up front, Texas, and the gentleman in the red sweater. Those were the students who made the bacterial photography system. One of the 5 projects worked that third time round. And so that was so much fun that we taught it with 13 schools, 3 outside the US. And then we taught it at 36 schools the next year. And the students now were sort of in control, meaning they made a world map distribution showing where the teams were. And they noted, for example, that nobody was from Australia, very few people from the Southern Hemisphere, nobody from China. As a course of their normal exchange programs, a bunch of the students were over in China, for example. They visited the top 6 universities in China. And those students then decided to have a workshop so they could learn how to be competitive in this competition. And next thing you know, we're over in Tianjin, giving a 2-day workshop talking about this sort of work. And 5 of these universities showed up at MIT the next year as participants, and the team from Peking University in Beijing won. And we're doing it again, so we have about 1,000 students showing up November 8th and 9th at MIT to present their work in this year's an International Genetically Engineered Machines competition. It's sort of a genetic engineering Olympics. I'll end with 2 slides and a question slide. So this is the project from University of California, San Francisco. And I show it because UCSF is a very prominent medical school, and they don't have undergraduates. And so the team there was assembled from high school students at Lincoln High School, which is a public high school in San Francisco. And I kid you not, these folks showed up at MIT last November, and they blew me away. They came up with the following project. They said, you know, when we write a genetic program and we put it inside a cell, oftentimes it doesn't work. And we don't know why it doesn't work. We don't know if it's because our design was really bad, or we don't know if when we put the DNA program into the cell, you get all sorts of weird interactions. And we'd like to separate these 2 types of failure. And our inspiration here actually comes from computer programming. How many folks have heard of a programming language called Java? A bunch of people. So what makes Java really cool is you can write a Java program and it'll run on many different types of computer operating systems and hardware. That's not magical. The reason that's true is there's some underlying tool. It's called the Java Virtual Machine. And the Java Virtual Machine is built on every major hardware and operating system. So an Apple or a Dell or what have you. And the Java Virtual Machine then provides a common operating environment for the Java program that you write. And so the same Java program can run on a Dell machine or an Apple machine or a Linux box. Does that make sense? So you need this Java virtual machine. Well, they set out to make a virtual machine for genetic engineering, for DNA programs. And the way this would work is obviously you would boot up basically a little compartment, a little organelle inside whatever organism you wanted to run your DNA program within. And this synthetic organelle, which they called the synthesome, would become the equivalent of a virtual machine for synthetic biology. These are high school students. Pretty cool. How would you do this? Well, it turns out that in this case in yeast, there are receptors that sense signals in the environment. And when they sense signals, the receptors get sucked into the cell and destroyed. And this is a mechanism for resetting the sensing machinery so that you don't keep responding to a signal that was an old signal. They figured out that you could redirect receptor endocytosis and destruction by modifying a phospholipid on the vesicle, pretty sophisticated biochemistry, it turns out. And then they added in a new phospholipid from humans that provides independent addressing, such that over the period of a summer, they were able to prototype a system where before or after the presence of a signal, you can see very few little volumes and the beginnings to have clusters of a new, what's hoped to be a synthetic organelle. Summer term project for high school students. So to end, here's some questions. These questions are meant to be illustrative. This one illustrates issues of safety, perhaps. Here's a question related to security. The United States, for example, we had an active bioweapons program from pre-World War II to the Nixon administration. What would happen if we took the new tools that are being developed and deployed them to cause harm? Will we have biohackers, and will they be good or bad? Should these parts, they be owned by me, by MIT, free to use? What would be equitable? Bridge designers, we have to sign our designs if we're doing works that impact the public. It's surprising to me that after 35 years of genetic engineering, we don't have the American Society of Genetic Engineers. We don't have professional practices. Should we be doing this? I don't know exactly what the answers are around these sorts of questions. I have strong opinions. But my strongest opinion would be because the technologies that are driving this are changing so dramatically, we probably need to prepare to have constructive conversations about this for decades to come. Thank you again for the invitation and your patience. I'm happy to stick around and take questions.
So we have a couple of microphone runners. This is off. We're going to bring microphones around. If you have a question, would you mind raising your hand, please? Sure. Joe back.
As a mentor for hundreds, thousands of high school students, what do you tell them about the ethical implications of the work that they're doing and what to look out for? You implied you had some thoughts on it, but I'd like to hear them.
Yeah, so in order to be a competitor in iGEM, the Genetic Engineering Olympics, you have to answer, as a student, a series of questions about safety. You have to do a self-assessment to consider whether or not you think there are any safety issues related to your project. You then have to ask people at your local institution who are in charge of biological safety what they think about your answer and your project. You then have to provide information about any of the new biobrick parts, the genetic componentry, such that other people who might choose to use them are made well aware of such issues. And so with respect to biosafety, what we're doing is extending and hopefully complementing the last 35 years of relatively strong success around recombinant DNA technology. Now, I want to be careful there because you can evaluate safety from three perspectives at least. One is the safety of the researcher, or the student in this case. The second is the safety of the public, all of us outside their labs. And the third might be the safety of the environment, right? And so the frameworks I'm thinking about have mostly to do with researcher or student and public safety. And so none of these projects are meant to be, in this context, released into the environment, which would trigger a separate set of concerns and review. So one thing we do, in answer to your question, is we're very prescriptive. We insist that they and their community directly consider issues of safety. Practically, what we're dealing with are very significant challenges. The first challenge is a generational transition. When recombinant DNA was invented in the 1970s and brought into the then existing biosafety framework, well, that was just a long time ago. Our students were not alive then. So there's a generational transfer of wisdom. The second practical challenge we're dealing with is most of these students are not microbiologists. Not that there's anything wrong with microbiologists, we need more of them. They tend, in this case, to come from computer science or physics or civil engineering, for good's sake. So they don't have the same, cultural background that comes from getting a major in biology necessarily. So we're dealing with a generational and cultural transition around issues of safety and passing on existing and past wisdom. And our approach so far has been to be, as I mentioned, prescriptive. There's a second aspect of this which has to do with the architecture of the competition itself. When the competition was first proposed, and almost every year, invariably, somebody suggests that the competition be called not iGEM, and the end of competition event is called the iGEM Jamboree. But instead, they suggest it be called Bug Wars, BUGWARZ, or something like that. Because obviously, this would be more exciting. Teams of students will work to engineer, faster growing viruses or bacteria. They'll all show up, they'll put all their critters into a bucket, and we'll go out for a pint, and we'll come back and see who has the highest titer. That would be a really bad idea, right? So at a very practical operational level, we have prescriptive mechanisms in place. And at an architectural level, by operating this event, we're occupying a space in the ecology of education that might be occupied by something else. I hope that that's sufficient, but we'll see. I think there are probably other things that we could be doing and need to consider, and I'd welcome any input on that.
Thank you for a very, very inspirational lecture. The parallels with computers and computer languages, and indeed languages in general, and grammars in general, are wonderful. And the engineering sense of it is wonderful. Three things occur to me that need some sort of special handling, and I'm wondering about the extent to which you've contemplated them. One is that biological things, if you will, have three natural propensities that we know of. One is that of self-assembly, another is that of reproduction, and another is that of mutation. Those three things are kind of wild cards in your dictionary of tools. How do you propose to, or have you, or are you contemplating addressing those issues, especially with high school students trying to construct organisms?
Self-assembly, reproduction, mutation. Let me add a fourth. These things operate on very small physical length scales. And so as soon as you start talking about individual reaction events, it's not necessarily the case that you're going to get reliable deterministic behavior. You can get stochastic behavior. And so these are four physical problems associated with engineering biology. We don't have a theory for design of reproducing machines, would be to answer one of yours. In part, I think that's because engineers haven't had reproducing machines to work with. Von Neumann, ironically, at the end of his life, is working on a theory of reproducing automata. And there's a book which he started and somebody else completed on that topic. But it's more empirical than a solution. The way I think about that particular issue, at least as a starting point, and I don't have an answer, but I'm curious about mapping machine reproduction, or I call a cell a machine, but whatever, into information theoretic framework, where the parental generation is the transmitter or the sender. The offspring or the children are the receiver, sender and transmitter and receiver of a message. The message that's being communicated is the design of the machine, and the channel for communicating the message is the means of reproduction. And if we can make that mapping, then it might become interesting to begin to understand the mechanisms for error detection and correction. And at a very high level, that's about as far as we've taken it. I'll simply note that if we can't figure out how to design understandable reproducing machines, then we might, as a practical surrogate, make non-reproducing, disposable biological machines. And if you understand the significance of DNA synthesis chemistry, then that becomes a possibility. Whether it's energetically or economically affordable is a different question. In terms of reliable behavior, the fourth one I added is real interesting. And there's been a lot of work happening on that over the last 10 years. Most of it has pointed to the fact that biological systems at the molecular and atomic scale are unbelievably noisy. This is yet to be reconciled with the fact that much of biology we're familiar with is reliable. And so we have to figure out, and I don't know exactly how this is going to play out, whether or not we can make reliable components from unreliable parts. Right?
Mutation.
Mutation. So I think we've got a lot of room to work there. For example, could you imagine designing a genome for which every mutation is deleterious? Many mutations today are tolerated, right, in natural DNA sequences. And when folks look at the natural genetic code, they see that some mutations have effects, but many mutations don't have effects. So as a thought experiment, could you design a genetic code for which every mutation is deleterious? This is what an engineer would call a fail-fast genetic code. As soon as anything changes, shut the whole thing down because it's broken. Well, it turns out you can certainly think about that and come up with an idea. The idea would be to expand the genetic code. Normally there's 3 bases of DNA for every amino acid making a protein. You would expand the genetic code. Instead of it being 3 bases per amino acid, you'd make it 4 bases per amino acid. But you'd still only have 20 amino acids. And you'd only use 20 of the 256 4-base codons. I'm sorry this is a little bit technical, but just to give you a specific example. What this would mean is you'd have enough empty codons such that you could surround every coding codon with a null that coded for nothing, right? So and oh, by the way, Pete Schultz at Scripps has made a 4-base anti-codon pair and shown that it incorporates amino acids into proteins. Now this would be an incredible genome re-engineering project of the sort of which I haven't ever seen proposed except just as a thought experiment. But you might be able to make fail-fast genomes, right? So to the extent that natural biological systems are selected to be evolvable, we as naive biological engineers might reprogram them to be different. And then also on this topic, I didn't go through this, but on this data sheet, in the meantime, since we haven't made that fail-fast genome, you'll see here, this is stolen from something that Texas Instruments made. This whole architecture, this data sheet, this is from the Transistor Logic Data Book, which is a book of thousands of pages about electrical components. Something that's interesting on this, which isn't found in the Transistor Logic Data Book, is this plot right here. This is showing how this genetic device will perform over 100 generations under different environmental conditions. So in condition A, there's no input signal. And what we find is the performance of this device and the genetic material is stable. It sticks around as far as we looked, 100 generations. Under conditions B, where we're driving the device with a strong input signal, it mutates. Consistently, it goes away after 72 doublings. And the reason it's consistently going away is the burden of this device on the cell is so much that mutations are arising very, very quickly. They arise, relatively speaking, instantly. And it takes 70 generations for the mutant to take over the population. So in the absence of being able to program evolution, we can at least begin to characterize it and study it and see where we go. Self-assembly, I showed you the DNA self-assembling systems. We have good practical success with 2 dimensions. There's a theoretical treatment in Saul Griffith's thesis from MIT a couple years back for 3-dimensional systems. I haven't seen that implemented. Machine reproduction, I don't know how to make a reproducing machine.
Thank you very much.
Another question?
Yeah.
Have you ever considered using the amino acids that are other than the 20 that are in nature? Because I read there are more than 20. I think they're 50 or 60.
Yeah. So there's a category of amino acids called unnatural amino acids that chemists and others are fond of. And they have pretty interesting properties. I've not worked on them myself, but I think if you look at the interface, in particular between biology and materials, you could use some of these unnatural amino acids to make interesting things, right? And I would point you again to the work of Peter Schultz at Scripps, if you're familiar with that. The challenge of a lot of the unnatural amino acids work, in my opinion, and the reason I think you're unlikely to see it incorporated into the student projects, is there aren't biosynthesis routes worked out for the production of these unnatural amino acids, meaning you have to add them separately. The cells can't make them themselves. And that's a big limitation.
So far.
So far. Yeah, fair enough. So that'd be a good project to work on, right, if you cared about such things.
One more question? On the back center.
in a sense, okay. So in a sense, you said the evolution route has given us these things that work really well, but we can't understand them. Is there any thought about tapping some of the power of evolution in some of these evolved systems? I know you got a circuit diagram that at least one person didn't like, but can you start with a machine that works okay, run it through some evolution and see, wow, we have a better one and I didn't think of that?
That's a great question, and not something I do a good job of being clear about in the talk. Obviously, most of the componentry we're starting with comes from evolution or nature, right? So we benefit greatly from that to begin with. Then you note, if you make something that kind of works, could you then tune it up by mutating and selecting it in the laboratory? And the answer is absolutely yes. And so Yohei Yokobayashi at University of California, Davis, and Francis Arnold at Caltech have both had tremendous success in pioneering the application of evolution to begin to tune up some of our engineered devices. The challenge is to figure out how to get better at programming evolution, right, and implementing more sophisticated screens and selections in a laboratory setting, and then combining that with the levels of abstraction shown here, so that you could imagine applying evolution at each level in a composable hierarchy that also supports forward engineering. All right, so one way to think about it is you yourself might be the author of a genetic program, but evolution might be your editor, as Francis Arnold would say. It's a good question.
I think we'll end there. And I just want to remind everyone in the audience to please complete your surveys that you received when you walked in. Those are important to us. We really like to get your comments and feedback as far as topics and all that kind of stuff. And that was tonight. So